Introduction. Welcome, medical students, to today's lecture on the fascinating topic of ketone body metabolism. Ketone bodies are small, water-soluble molecules produced in the liver during periods of fasting or carbohydrate restriction. In this comprehensive script, we will explore the intricate regulation, function, and implications of ketone bodies in various physiological and pathological contexts. Let's dive into the details of this intriguing metabolic pathway. During prolonged periods of fasting or starvation, the body's glucose reserves become depleted, necessitating alternative energy sources. This is where ketone body metabolism comes into play. The liver enzymatically converts fatty acids into ketone bodies, such as beta-hydroxybutyrate, BHB, acetoacetate, ACAC, and acetone. The rate-limiting step of this process is the mitochondrial enzyme, HMG-CoA synthase 2. Its expression and activity are tightly regulated by hormonal and nutritional cues, including glucagon, insulin, and free fatty acid levels. Additionally, the transcriptional regulator PAR-alpha plays a crucial role in the upregulation of key ketogenic enzymes, such as fatty acid beta oxidation enzymes and ketone body metabolizing enzymes. The brain, although primarily reliant on glucose as its energy substrate, has a remarkable ability to utilize ketone bodies during times of glucose scarcity. Ketone bodies can easily cross the blood-brain barrier and serve as an efficient energy source for neurons. Beta-hydroxybutyrate, in particular, exerts neuroprotective effects by promoting neuronal stability and reducing oxidative stress. The brain's transition to using ketone bodies as fuel is facilitated by the upregulation of monocarboxylate transporters, MCTs, on the blood-brain barrier and neuronal plasma membrane. These transporters facilitate the transport of ketone bodies into the brain. Epigenetic modifications, including DNA methylation and histone acetylation, play a crucial role in the regulation of key ketogenic enzymes involved in ketone body metabolism. For instance, studies have shown that DNA methylation events occurring in the promoter regions of genes encoding ketogenic enzymes can modulate their expression levels. Histone acetylation, on the other hand, can loosen the chromatin structure, allowing for enhanced transcription of these genes. Understanding the epigenetic regulation of ketone body metabolism offers potential therapeutic targets for metabolic disorders. Mitochondria, the powerhouse of the cell, play a pivotal role in ketone body metabolism. Ketone bodies are transported into mitochondria, followed by their conversion into AC-CoA in a series of enzymatic reactions. This AC-CoA is further utilized in the tricarboxylic acid cycle, TCA, to generate energy in the form of ATP. Importantly, the utilization of ketone bodies as an energy source promotes mitochondrial biogenesis and enhances oxidative phosphorylation efficiency. Additionally, the production of ketone bodies generates NAD and FOD2, critical cofactors for ATP production via oxidative phosphorylation. Insulin, a hormone secreted by the pancreas, plays a crucial role in regulating glucose and lipid metabolism. Interestingly, ketone bodies have been shown to modulate insulin sensitivity and signaling pathways. High levels of ketone bodies can inhibit insulin signaling through mechanisms such as the activation of protein kinase C and the modulation of insulin receptor substrate 1 phosphorylation. Furthermore, ketone bodies themselves can act as signaling molecules, influencing cellular processes beyond their metabolic role. The proper transport of ketone bodies across various membranes is essential for their metabolism and utilization. The monocarboxylate transporters, MCT1 and MCT2, facilitate the transport of ketone bodies across the plasma membrane of hepatocytes and peripheral tissues, respectively. Disruptions in the expression or function of these transporters can contribute to metabolic disorders, including ketone body utilization defects. Future research may shed light on therapeutic interventions targeting these transporters to restore ketone body homeostasis. Cancer cells exhibit altered metabolism termed the Warburg effect, characterized by increased glucose uptake and reliance on aerobic glycolysis. However, recent studies have highlighted the significance of ketone body metabolism in cancer cells. Ketone bodies can serve as an additional energy source and provide intermediates for biosynthetic processes in tumor cells. Understanding the specific metabolic alterations in cancer cells may unveil potential therapeutic strategies targeting this unique metabolic feature. Several neurological disorders, including epilepsy and Alzheimer's disease, have shown associations with impaired glucose metabolism. Interestingly, emerging evidence suggests that the ketogenic diet, which elevates ketone body levels, can provide therapeutic benefits for these disorders. Ketone bodies act as alternative energy sources and modulate neurotransmitter release, neuronal excitability, and inflammation, thereby potentially ameliorating the pathophysiological processes underlying these neurological conditions. Sepsis, a life-threatening condition resulting from a dysregulated immune response to infection, is associated with significant metabolic alterations. Studies have shown that ketone body metabolism is perturbed during sepsis, highlighting its potential role as a biomarker in disease progression. Furthermore, therapeutic strategies aimed at restoring healthy ketone body metabolism may hold promise for improving outcomes in septic patients. However, further research is needed to delineate the intricate relationship between ketone body metabolism and sepsis. Inflammation and immune responses are tightly interconnected with metabolic processes. Ketone bodies have been shown to modulate inflammatory signaling pathways and immune cell function. Specifically, ketone bodies exhibit anti-inflammatory properties by inhibiting the NLRP3 inflammasome, reducing pro-inflammatory cytokine production, and influencing immune cell migration and activation. The precise mechanisms through which ketone bodies regulate inflammation and immunity warrant further investigation. Ketone bodies are not only crucial for adult metabolism but also vital for the growth and development of the fetus and neonate. During pregnancy, ketone bodies serve as an essential energy substrate and contribute to the production of structural lipids in the developing fetus. Furthermore, neonates, particularly preterm infants, exhibit high circulating levels of ketone bodies due to hepatic immaturity. This endogenous ketosis provides an important energy supply during the transitional period before establishing regular feeding patterns. Exercise training induces numerous metabolic adaptations, including changes in ketone body metabolism. Prolonged endurance exercise promotes the utilization of ketone bodies in skeletal muscles, thereby preserving glycogen stores for more demanding exercise bouts. Additionally, exercise training increases the expression of key ketogenic enzymes and transporters involved in ketone body metabolism, optimizing their utilization for energy production. Understanding exercise-induced adaptations of ketone body metabolism may have implications for athletic performance and metabolic health. In conclusion, the study of ketone body metabolism has revealed remarkable molecular, cellular, and physiological insights. From the regulation of ketogenic enzymes to their impact on various organ systems, ketone bodies have emerged as versatile molecules with diverse functions beyond their role as alternative energy sources. Further research into the intricacies of ketone body metabolism promises to uncover novel therapeutic interventions for metabolic disorders, neurological conditions, and critical illnesses. As future physicians, your understanding of these complex mechanisms will be invaluable in providing optimal care for patients.